Welcome. My name is Sharon Mitten and I'm Head of Fundraising and Philanthropy here at the Florey. Before we begin, I would like to take a moment to reflect on the meaning of place, and in doing so, acknowledge the traditional owners of country and where we are today. The Florey Institute is on the land of the Wurundjeri people of the Kulin Nation, and I'd like to pay respects to the elders past, present and emerging. We celebrate the traditional owners as the first scientists and deeply respect their sophisticated knowledge systems. For those attending who are not familiar with our work, the Florey is the largest brain research group in the Southern Hemisphere. Our work is critical as diseases of the brain and mind affect all of us in some way. In fact, every year 4.7 million Australians are diagnosed with one of the conditions we are studying. This year and the last year have highlighted the importance and need of our work more than any other since our very beginnings. Topics which we live and breathe daily as a medical research institute have been under the spotlight and in the public sphere. Thank you to all our donors who have joined us this morning. As a medical research institute, our work relies heavily on your support. Ways you can also support our research is by spreading the word about the Florey and the learnings you take away from the lecture today. If you're not already a Florey donor, we invite you to consider becoming one. All contributions, big and small, are tax deductible and take us one step closer in our quest to find the cures for diseases that affect the brain and mind. You can also support our work by leaving a gift in your will. If this is something you would like to consider, please email the fundraising team after the lecture. We'd also love to keep you up to date with the research happening at the Florey and invite you to join our mailing list if you're not already a subscriber. Please head to the Florey website. I'd now like to introduce our presenter today. Dr. Leigh Walker is an early career researcher here at the Florey. She has completed a Bachelor of Biomedical Science with honours in 2013 at the Victoria University of Wellington, New Zealand, and her PhD at the University of Melbourne in 2018. Lee's current research focuses on discovery and development of novel targets to treat alcohol use disorder. Specifically, she's interested in the link between stress and excessive alcohol consumption and seeking and using pharmacological genetic tools to map and manipulate specific neural circuits underpinning aberrant behaviour. In 2020, she was awarded the NHMRC Ideas Grant to pursue research examining sex differences in stress and alcohol interactions. I've been privileged to have a sneak peek at today's presentation, and I'm really excited to be sharing it with you all today. Over to you, Leigh.
Thank you all for coming today. And I would also just like to start by acknowledging the traditional owners from where I'm speaking to you today, the Wurundjeri people. And I'd like to pay my respects to their elders past, present and emerging, including any Aboriginal or Torres Strait Islanders people who might be joining us today. So as Sharon said, my name is Leigh Walker and I'm a researcher here at the Florey Institute of Neuroscience and Mental Health. And today I'm going to talk about why alcohol and stress don't mix. So I'll start off with some background and then share some work that some of the findings that we have from our lab. When we compare ourselves to the rest of the world, Australians are some of the highest consumers of alcohol. So this image here shows per capita consumption of alcohol in litres for adults. And as you can see, Australians are well above the average here. And not only is it how much we're drinking, it's how we drink. So data from the Australian Drug Foundation, Alcohol and Drug Foundation, has shown that alcohol is the most common and widely used drug in Australia, and that 1 in 5 Australians over the age of 14 drink at levels that put them at risk of harm and developing an alcohol use disorder. So not only that, data from the World Health Organization has shown that 4.4% of Australians have an alcohol use disorder or have had an alcohol use disorder, and 6.1% of those are men and 2.7% women. So this is over 1 million Australians throughout the country. So alcohol also impacts society. So in Australia, it leads to 430 hospitalizations every day and kills 15 Australians every day. And on top of that, costs the Australian economy $15.3 billion every year. And these are statistics from the Foundation of Alcohol Research and Education. So alcohol, harmful use of alcohol is also implicated in over 200 different diseases and health conditions. So it contributes to 48% of liver cirrhosis. It also contributes to mouth cancers, pancreatitis, tuberculosis, other types of cancer, colorectal and breast, and also hypertensive heart disease. But not only that, it also has other implications, obviously leading to alcohol use disorders, but also implicated in suicides, interpersonal violence and domestic violence, and also a large number of traffic injuries and epilepsy. So alcohol is actually the #1 leading cause of preventable death in people aged between 15 and 49 worldwide. And alcohol leads to over 3 million deaths every year. So that's 6 deaths every minute. So when I talk about alcohol use disorders, what am I actually talking about? So drinking is a continuum. Like many diseases and disorders, there's a spectrum. And at one end, you have the safe level of drinking. So this is based off the number of drinks that you consume. So for men, this is a max of 4 in a day or in a session, and up to 14 per week. And for females, it's a max of 3 in a session and up to 7 per week. And then you move into at-risk drinking. So that's just drinking volumes that are greater than the safe ones. And then next is what we consider problem drinking. So this is when it's not just about how much you're drinking, but there's it begins to impact your life and your lifestyle. And then there's this imaginary line where you transition into an alcohol use disorder. So again, within alcohol use disorders, there's a spectrum. So from mild through to severe. And this isn't based on the number of drinks that you consume, but it's actually how alcohol is impacting your life. So to determine whether somebody has an alcohol use disorder or the severity of the alcohol use disorder, people answer a questionnaire. So this is widely available online. Anyone can look it up. It's called the DSM-5, or the Diagnostic and Statistical Manual, and we're up to the 5th edition. And what it does is it asks 11 different questions about your relationship with alcohol and its impact on your relationship in your work and your general lifestyle. So there's different types of questions on here. So, you know, more than once, have you wanted to cut down or stop drinking and tried to, but you couldn't? Or a new one to the DSM-5, you know, have you wanted to drink so badly that you couldn't think of anything else? So sort of the craving of alcohol, or, you know, has continued drinking, have you continued to drink even though it's causing trouble with your family or your friends, so it's starting to impact your relationships? Or, you know, have you given up or cut back activities that you used to find pleasurable or important in order to drink? So prioritizing drinking over other things that used to give you pleasure. So for a mild alcohol use disorder, you would be considered if you answered yes to 2 to 3 of those different 11 questions. Moderate, considered if you answered yes to 4 to 5. And severe, 6 or more of those symptoms. And so, as I mentioned, alcohol use disorders are really common. It's actually very likely that everyone here today has either suffers from or has suffered from problems with alcohol, or knows someone close to them who does, you know, a mother or a father or brother or sister or a close relative. And globally, 7.6 million people, you know, suffer from alcohol use disorders. And as I mentioned, over 1 million Australians. And addiction and alcohol use disorders are really highly stigmatized. And that means that a lot of people actually don't seek help. So less than 20 people actually seek help, and less than 5% of those actually, or 5% of people get treated with different medications. So if we look at what the current medications are that are available, the first one that was approved is called disulfiram or antabuse, and this was approved in the 80s. And what it does is it just interferes with alcohol metabolism. So if you're taking this drug and then you drink alcohol, it makes you feel ill, and it could cause hot flushes or nausea and things like that. But there's a few problems. The first is that it's not actually PBS approved, so it's very expensive in Australia. And if somebody wanted to drink, they can just not take the drug. So there's low compliance to taking the drug. The next one approved was naltrexone, and this is an opioid receptor antagonist. So what it does, it just interferes or blocks the high gained from alcohol use. And it's somewhat effective in about 1 in 10 people, but it's more effective in people who have a certain mutation. And the last one is acamprosate or Camprol, which was approved in 2004, but we don't actually know how this works. We know that just in some people that it prevents relapse or helps people to maintain their abstinence. But as you can see, you know, no new pharmacotherapies or no new treatment medication options to treat alcohol use disorder have been, you know, approved since 2004. And at a population level, all of the treatment options that we currently have are inadequate. So they all suffer from either reverse side effects, low compliance, high rates of relapse. And this really underpins why we need to understand what's happening in the brain so that we can try and develop more effective treatment options that can treat more broadly across the spectrums of alcohol use disorders. So what causes alcohol use disorders? You know, there is no single cause. The slide is busy, and that is because it is a very complex behavioral and neurobiological disorder. So there's several key factors that can influence it. The first is alcohol itself. So, you know, if you never drink, then you can't develop an alcohol use disorder. But there's also significant contributions from both biological and environmental factors that contribute. So biological factors such as your genetics. So it's thought that genetic factors contribute to up to 50% of alcohol use disorders. You also have your biological sex. So different sex hormones like estrogen and testosterone can contribute to different drinking patterns. Circadian rhythm, so how you're sleeping, your sleep patterns, and different comorbid diseases. People with anxiety, depression, and other neuropsychiatric disorders have a higher vulnerability to develop an alcohol use disorder. And then we also have environmental factors such as your social interactions, the groups, friends that you interact with, culture and religion, whether that be sporting culture as well. And then, you know, context and cues, where you hang out. Do you hang out at places that serve alcohol a lot? Stress, which is something that I'll talk a lot more about as we go through. And abuse and neglect is another one. So up to 70% of your risk of developing an alcohol use disorder if you've had childhood neglect or abuse, it's 7 times higher that you would develop an alcohol use disorder. So all of these different factors can really influence. And so now I'll briefly give you an overview of sort of how the chemicals in our brain signal with each other, or how brain cells communicate, so we can try and understand when I explain the rest of this talk. So what we have here is a brain cell. called a neuron. And you can kind of think about these as telephone wires that connect one part of the brain, allowing messages to travel to another part of the brain. So all drugs of abuse work roughly in the same way, where they cause release of a chemical messenger. In this case, this image shows dopamine. And then what happens is this, leads to a feeling of pleasure by binding here to a receptor, and results in communication from one cell to another cell. And so how does alcohol actually hijack the brain? So what it does is it acts on the natural reward circuitry, or the natural reward pathway. So this is really necessary for survival. So, eating, procreating, etc. So they're all feel-good feelings, and they're positively reinforced, so therefore that you repeat them. So alcohol, causes a rapid surge of dopamine and other feel-good chemicals in the brain. And if this is repeated, what you actually end up getting is a reduction in these feel-good chemicals produced in the brain, and an upregulation in different stress chemicals in the brain. And then if you have prolonged alcohol abuse, where you keep these binge cycles happening, you're acting on a brain that already has a decrease in the number of feel-good chemicals and an increase in the stress chemicals. So what it leads to is you need more alcohol to get that feel-good. So this is what we call tolerance. You've built tolerance to the alcohol. You need to drink more. And also it leads to, a change where things that were previously pleasurable are no longer pleasurable. You have cravings, and these cravings are 3 to 5 times stronger than for natural rewards. So if you think about when you're hungry and you want to eat, how much you crave food, think about 3 to 5 times that amount. That's what people with an alcohol use disorder can crave for alcohol. And then you also have withdrawal symptoms. So when you stop drinking, which leads to this negative reinforcement to drive drinking. And so another way to think about this is as a cycle. So addictions, including alcohol use disorder, are characterized as a chronic relapsing nature. So where you have repeated alcohol consumption, then you go through a period of abstinence, and then you relapse. So after the pleasurable part, which is driven by this dopamine release in the basal ganglia, so parts of the brain that you might have heard of, the striatum and the nucleus accumbens. So alcohol becomes sort of more of an ingrained behavior, where the impulsivity shifts to compulsivity, and the primary drivers of repeated alcohol consumption shift from positive reinforcement, so the positive feelings, the feelings of pleasure, to negative reinforcement, or a feeling of relief. So what happens is, as the person, seeks to stop the negative feelings, so these might be things like regret, guilt, shame, and also the physical illness that's associated with the withdrawal symptoms, that you have part of the brain called the amygdala that starts to drive this alcohol consumption. So instead of it being driven by the positive feelings of drinking, now it's all about escaping the negative feelings. And that's a stronger reinforcing thing in the brain than the positive feelings. So the last stage of this addiction cycle is controlled by the decision-making part of the brain, or the prefrontal cortex, which is involved in executive control. So this is the last part of our brain that fully develops. It doesn't develop until we're a bit older, about 25. And so this is why we sometimes don't make the best choices in our late teens and early 20s. And so then this, is what causes the craving, and then the decision to relapse, or to consume the drug. So it sort of comes down to this balance between the control part of your brain trying to stop you from relapsing, but these negative emotions sort of overriding your sensible brain, and causing relapse. And it's not a matter of sheer will. You can't just sheer will yourself a lot of these times, because the brain has actually changed and the cards are really stacked against you. So it makes it really hard to stay abstinent. And one of the biggest, things that can contribute to this is stress and anxiety. So a recent study by the Australian Bureau of Statistics in 2018 showed that 2.4 million Australians have high or very high psychological distress, and 3.2 million Australians had an anxiety-related condition. And so we know that stress and anxiety disorders are also highly comorbid with alcohol consumption. So 2/3 of people who suffer from distress or anxiety drink alcohol. And it's been reported that 40% of Australians use alcohol as a coping mechanism for stress. And these rates are increasing. So even in the years from 2015 to 2018, we saw a 10% increase in the rates of distress and anxiety of Australian adults. And that was even before we've had to contend with the last few years. So it's continuing to rise into 2020 and 2021, which, is unexpected, is very expected. Considering, over the last 18 months, our society has been severely impacted in one of the fastest and most profound ways. So Australians have had to isolate, and we've had to change our way of life. And especially those who are in Victoria, so we're just, easing out of our 4th lockdown, and people have had to contend with lost job security, financial strains, isolation, and worry about loved ones. A lot of people have had to worry about the stress of homeschooling and working from home. And on top of that our own, physical and mental health. And a recent study from America, from the American Psychological Association, examined pandemic stress 1 year on in a vulnerable population. So that's parents who were, in America. And what they found was that many of them really didn't feel like they got the support that they needed. And nearly a quarter were diagnosed with a mental health disorder in the last year of the pandemic. And this has gone on, to impact other factors of their life, including their sleeping cycles, how they're sleeping, different weight changes, gaining or losing weight, and also how they're drinking to cope with stress. So between 30 and nearly 50%, so 30% of females, up to nearly 50% of men, were drinking more to cope with stress. And so, we've all been exposed to a lot of these headlines that have really been highlighting the worry about drinking and how Australians are drinking during this pandemic. And there's been a recent study, or there was a study during the second lockdown by the Foundation of Alcohol Research and Education. And what they found is really alarming. So they saw that 20% of households bought more alcohol than usual since the COVID-19 outbreak. 70% of people who drink reported that they were drinking more than usual. 34% of people were now drinking daily. And 32% were concerned with the amount of alcohol that either they or someone in their household was drinking. And again, we see this link with stress, with 28% of people reporting that they were drinking more to cope with anxiety and stress. And so why does stress and alcohol not mix? Well, it's really ironic, isn't it? Because, although alcohol initially helps us to relax, what happens is over time it actually creates more anxiety. And there's a really strong interaction that goes both ways between stress and alcohol. So stress, we know that it increases alcohol consumption, and alcohol is used as a coping strategy for stress. We know that stress alters the rewarding properties of alcohol, so it makes it subjectively more pleasurable. And we also know that alcohol dysregulates the stress system in the brain. And as I mentioned before, stress is a major trigger of relapse, relapse or return to drinking after being abstinent for a period of time. And this is one of the biggest hurdles for us in trying to develop treatment options or drugs to help people to stay abstinent from alcohol. With 90% of people who suffer from alcohol use disorders relapsing within a year of abstinence, and the majority of those are within the first 3 months. And so what I'm interested in, and what my research is really interested in, is the neurocircuitry and the different neurotransmitters. So the neurocircuitry being the different brain regions and how brain regions are connected, and neurotransmitters being the chemicals that communicate between those brain cells. And I'm really interested in understanding how they're involved in this relationship between stress and alcohol. And I'm especially interested in a part of the brain called the amygdala, which is part of the limbic system. So it's located deep down in the brain here. And it's connected with both emotional and behavioral functions. So it's responsible for those really strong reactions, fear, anger, and sexual behaviors. And so what happens is when you have low to moderate stress, it's actually can be good for you. And what happens is you have the prefrontal cortex, that decision-making part of your brain, or your executive control center, and it dampens the amygdala activity by sending messages about whether, or the pros and the cons about a different reaction to something. But what happens when you have high stress is what's called this amygdala hijack. So the amygdala actually overrides the prefrontal cortex, and there's a heightened response. And so, we know that the amygdala changes in the brains of people with alcohol use disorders. So this is an example of a study where they used fMRI, so a brain imaging technique. And what they can do is they can scan the brain to detect the changes in brain activity when people are looking at something. So the more red something appears, the more active the brain region is. So what they did in this study was they showed people with alcohol use disorders 2 different types of cues. They showed them alcohol-related cues, and then they showed them neutral cues, which are just, in this case, a soft drink. And they measured activity throughout the brain. And so when they looked in the nucleus accumbens, so that's part of the brain regions that are involved in the reward, the dopamine signaling, and the feel-good parts, they didn't see any significant difference. They didn't see any difference in the brain activity to alcohol or soft drink cues. But when they looked in the amygdala, they saw this really big activation specifically for alcohol cues. So not the same for the soft drink cues. So what we can see here is that this brain region is having a really strong reaction to alcohol. And when they asked the people with alcohol use disorder in this study how much they were craving alcohol, they saw that this activation actually correlated or it was associated with how much craving they were feeling. But there's actually, a few limitations to using MRI scans. So it's just these random blobs. What do these blobs actually mean? What's the causal relationship between this brain region and alcohol and stress? So to do this, we can use animal models. And what's really important about our animal models is that we want to make sure that they reflect the same changes that we see in the human brain in our animal model. So how similar are we to the animals that we study? So we know that the homology or the similarity between humans and primates is really high, 98%. But what about a mouse? So we actually share 96% similarities of our genetic makeup with mice and rats. And most importantly, the brain is structured and organized in a similar way. And so this is really fortunate for addiction researchers, as the natural reward system that we're really interested in, and that alcohol hijacks, is similar in rodents and in humans. And this is because it's a really primitive part of the brain that's really important for survival. So it's conserved across different species through evolution. And so how do we examine alcohol consumption in our rats? So the rats are group housed, and then for 20 minutes a day, they get separated and taken to the bar. And so in the bar, they're placed into this chamber where they have the option to voluntarily consume alcohol. So there's two levers, and one, if they press it, they get alcohol, and the other one, if they press it, they just get a water. And so they get to make the decision about whether they'd like to get a water or if they'd like to get alcohol. So this is just a quick little video showing that. So we see the rat here, he presses the lever, we see this light illuminate, and what that is telling the rat is that there is alcohol available for him to drink in this receptor. And so we know that this rat really likes alcohol because his hand never leaves the bar. And what we can see over time, or different days, so they have 20 minutes per day access before they return back to their group housing. And what you see is that they learn to press this lever over time. And so after a period of time, this becomes stable. And then what we can do is we can place the rats into what's called extinction. So extinction is our equivalent of putting a rat into rehab. So here they still go into the chamber, but when they press the lever, they no longer get the alcohol delivered. And so now they learn that the lever no longer delivers alcohol, and they discontinue pressing the lever for alcohol. And so the next thing that we can do once they've sort of stopped pressing is that we can test their propensity or how vulnerable they are to relapse. So we use the same 3 factors that can cause relapse in humans. So a drug prime, so this might be having 1 sip of alcohol that leads to 3 or 4 beers, or a cue, which might be driving past your favorite bar or favorite bottle store, those things that sort of trigger you wanting to drink. And the last one is by making the animals a little bit stressed, just very mild stress. And what we can do then is put them back into their rat bar and see how many times they try and press that lever despite them not getting any alcohol. So that's our measure of how much they want alcohol. And so during my PhD, I was really interested to understand if the same parts of the brain that we know are active in humans are also active in rats after relapse that's caused by stress. And so to understand what parts of the brains are activated, we use a marker called CFOS. So this is our equivalent of a post-mortem functional fMRI scan. So CFOS is expressed in cells when they are activated. So we can use this as sort of a proxy marker to see which brain regions were activated by our behavior. So in this case, what cells in the amygdala are activated when a rat relapses because of stress. And so, what we found is that the same brain regions, we looked right throughout the brain, and the same 2 parts of the brain came up: the prefrontal cortex, that decision-making part of the brain, and the amygdala, or that stress, that reactivity part of the brain. And here on the right, show an image of what that brain looks like when we look at it under the microscope. So at the top, this is no relapse. So you can see the amygdala. There's these black dots that you see down the bottom here. these are a single activated neuron. So down the bottom, you can see there's lots of little black dots. Those are all different neurons that have been activated by this behavior. And up the top, we see a few, but not very many. So that's showing us that this brain region has lots of cells that are activated. So what makes us drink when we're stressed? So I know that my rat model of relapse activates the amygdala just like in humans. So next I wanted to understand what are the brain chemicals that might be released in the amygdala that might make us drink when we're stressed, or to relapse after we've stopped drinking. And so as I mentioned earlier, after repeat binge drinking cycles, there's this increase in these stress chemicals. And one of those that has been studied the most over the years is corticotropin-releasing factor, or CRF. So just a little timeline here. It was first discovered, 40 years ago, and then it was quickly found to be involved in stress and expressed in the amygdala, and has been extensively studied in alcohol use disorders using animal models. So this was a really obvious point for us to start at, to try and see if our cells that were activated in the amygdala were expressing CRF, suggesting then that they would release CRF. So to do this, what we do is we can stain for two different markers. So on the left here, you have the activated cells, so expressing CFOS. And then in the middle here, we've stained for a marker against the CRF neurons. So what we can do is we can look at where these overlap. So can we see overlap between the green here and the purple to show that those CRF cells are actually activated? But we actually didn't see very much overlap. So we quantified throughout a number of different brains, and we saw only about 2% of the CRF cells were actually activated. And CRF has been the most promising therapeutic target that's arisen from preclinical studies in the last 20 years. However, there've recently been 2 clinical trials looking at this in alcohol use disorder, and they've both failed to translate into positive outcomes in clinical trials. So this again highlights that, in our animal model where we don't see activation of the cell type, it aligns with the human clinical trial showing that it doesn't seem to be involved. And it also highlights that we need to look beyond the usual suspects. And so that's what I've been doing. And I've been looking at this neuropeptide or a neural chemical that is released called CART, or cocaine and amphetamine regulated transcript. So it's evolutionarily conserved between species, from zebrafish to humans, which is, as I mentioned, important to understand whether it could be important for reward-related behaviors, when we tested in rats and trying to move to humans. And it was discovered about 20 years ago, and it was named because of the fact that when they discovered it, both cocaine or amphetamine administration increased the expression in the brain. So it has nothing to do with cocaine or amphetamine. We're not changing what we're talking about. We're still talking about alcohol. That's just the name of it. So from now on, I'm just going to call it CART. And the thing is that, since that first discovery, it's been implicated in a bunch of other things as well, reward, feeding, and stress behaviors. And it's been linked to alcohol use disorder by the discovery of a mutation in the CART gene, so in the DNA of people, and that linked to a population of people who have severe alcohol use disorder in Korea. And I became really interested because it's really densely expressed in the amygdala. And we also know that it's expressed on different cells than the ones that express CRF. So here, CRF is shown in red, and CART is shown in green. And again, we don't see that this is the same cell that's expressing them. So are these cells activated by stress-induced relapse? And so to answer that question, I did this dual staining again. So here you see the activated cells, and in green you see the CART cells. And what we saw was, yes, we could see co-localization, or that these, were overlapping, the purple and the green. So this is telling us here that these CART cells in the amygdala are activated when an animal is relapsing because of stress, which is suggesting then that CART is going to be released in the amygdala. But again, just like an fMRI in humans, I'm just showing you blobs. What does it actually mean? So we can functionally test this in our animal models by inhibiting the CART chemical signal in the amygdala to see how an animal responds to relapse to alcohol. So to do this, we use our relapse test that I explained, so the same model. And on the left here in this graph, you can see lever pressing on the y-axis, or on this axis going up, and under no stress. So in the late stage of rehab, or what we call extinction, that they don't press a lot. But if the rats are stressed, we see that they significantly increase their responding, as we expect. And interestingly, if we inhibit CART chemical signaling in the amygdala, we can reduce their relapse behavior. So this isn't completely, but it's by about 50%. So showing that CART is involved in relapse caused by stress. So this was our first really interesting finding, that it could be really important here. So in science, once we have a finding, we want to work out what it actually means. So the next thing we wanted to understand was, does it influence relapse to other rewards like sugar? So this is an important question for us, because when we're looking to develop new drugs to target alcohol use disorders, we want to make sure that it doesn't influence natural reward consumption, only alcohol. So to do this, we train the rats in a similar way, but instead of pressing the lever for alcohol, they press for a liquid sugar solution. And what we can see here again is the rats with no stress, they have low lever pressing, and when they're stressed, they increase their responding the same way that they do for alcohol. But if we inhibit CART signaling in the amygdala, we don't see the same reduction. So this shows us that inhibiting this CART system seems to be specific to alcohol. And so then the next question that I wanted to understand was this because it's reducing the motivation to consume alcohol, or is it reducing anxiety behavior that leads to that increased consumption? So really, what is the mechanism that's underlying CART's ability to be able to reduce relapse caused by stress? And so to do this, we did two different things. So the first one was to measure the motivation to consume alcohol. So to do this, we put rats into the same chamber, but whereas before pressing the lever once led to one drop of alcohol, now we made it so that they had to work a lot harder, and it becomes exponentially hard for the rats to get alcohol. So for the first time, they press once. The second time they have to press twice, then 4, then 8, 16, 32 times, So the more motivated the rat is, the more they will press before they just give up. And we call this a break point. So when we blocked CART signaling in the amygdala, we didn't see a difference in the break point. So it suggests to us that it hasn't changed the motivation to try and consume alcohol. So the next thing that we looked at was the anxiety-like behavior using a light dark box test. So this is a measure of conflict in a rat, because they have a safe dark side where they're enclosed, but they also have this light side. And rats are really curious, and they want to explore a novel or a new environment, which is this light side. And so it's a conflict between wanting to be safe and explore a new environment. And the more times that rats spend in this light side, the less anxious they are. And so we had 3 different kinds of rats in this. So the first ones, they never had alcohol. And what we saw was when they were stressed, they still went out into the light side about 25% of the time. In equivalent rats that had been heavy drinkers, we saw a reduction in the amount of time that they spent on the light side, so showing an increase in their anxiety-like behavior. And when we inhibited CART signaling in rats that had been heavy drinkers, we saw an increase in the time that they spent in the light side, so back in line with the rats that had never had access to alcohol before. So suggesting that alcohol makes rats more anxious and that CART inhibition can restore this back in line with rats that have never had alcohol before. So just to summarize our data, so we identified a novel neurochemical or a novel brain chemical in the amygdala that reduces relapse to alcohol caused by stress. And this appears to be working by reducing anxiety caused by heavy alcohol consumption specifically. And so this is our first step to understand how this brain chemical could be used to treat alcohol use disorders. So what's next? So as Sharon mentioned, I was lucky enough to receive NHMRC funding last year to continue my research. So I'll specifically be focusing on sex differences in alcohol consumption. And this is a really important question to try and answer because we're seeing a really a strong increase in risky alcohol consumption and alcohol use disorders in women. So this is an old graph, but you know what we see here is the rates of risky drinking in men is pretty steady, but it's actually rising in women. So it increased about 80% in the last 15 years, compared to about a 30% increase in men. So that gap is really closing. And I'm especially interested because women also have are twice as likely to have an anxiety or a stress disorder, and also report drinking because of negative consequences, such as stress or anxiety, compared to men who often report drinking due to positive feelings or positive emotions like catching up with mates at the footy. And this area of research is also really important because there's been a real lack of research in female subjects. It's often been assumed that the female brain is just a small male brain. However, we really know that this is not the case. And there's a lot of, time and money that has been wasted in research and in drug development. One thing that I recently read was that in the last decade, 8 out of 10 prescription drugs in the US were withdrawn because they had greater health risks for women who were taking them. So not only is it a waste of money, time, resources, but it's also really dangerous for women. So it's something that I really want to try to understand. And so with that, I will finish, and I'd just like to finish by thanking my mentor, Professor Andrew Lawrence, and the people who were involved in the research that I presented today. And I'd also like to thank everyone here for listening. I hope that you have some more understanding about how alcohol use disorders aren't really just an issue of willpower, that stress and alcohol change the brain, and it has really huge impacts on the person suffering and for society really generally. So all of those costs to society, tens of billions of dollars every year. But despite this, addiction and alcohol research really don't get a lot of funding because of that stigma around it. Less than 1/10 of funding than cancer receives. So so I'll just leave you with that and open the floor for any questions.
We have got a couple of questions, Leigh. The first one is, does alcohol help with insomnia? I have taken sleeping tablets that are not effective.
So alcohol is actually, no, it's not very good for sleep. So maybe one drink will help you get to sleep quicker, but it actually, what it does is it disrupts your sleep during the night. So maybe you'll wake up more, you'll have a less restful sleep. So doing something like exercising or meditation, something to relax, getting rid of screens, something like that is actually more beneficial to how you're going to get a better night's sleep. But alcohol will definitely not help. It might be, again, like stress and anxiety, whereby you think it's helping in the short term, but it's actually having a more severe impact in the long term.
Yeah, the next question is about the rat experiment that you showed. And there's a person there, those, I suppose she talks about simulating real situations. And she says here, when the rats are put in the boxes, are they fed and hydrated? And do they have alternative choices other than alcohol?
Yes, so the rats, they always have access to food and water in their home cages. And when they're put into the chamber just for 20 minutes, they have access to either alcohol or water, but they're completely hydrated, they're completely fed. So it's completely voluntary whether they want to decide to press a lever for alcohol, or they can just sit there for 20 minutes. After 20 minutes, they'll just be taken out and put back, with their cage mates. Okay,
next one. Do you see evidence of a correlation between brain development in chronically stressed children? That means, for example, children that grow up in home care, and amygdala and frontal cortex function in relation to susceptibility to addiction?
So I don't personally look at that sort of thing, but I do know that there is definitely correlations between the neglect, child abuse, etc., in vulnerability to addiction. I don't know if there has been studies actually looking at how the brain is changed, but that is a really interesting avenue of research that, if it hasn't been done, should be done. I think that could, reveal some really interesting findings.
Another question there, also to do with children mostly. Historically, children are prevented from alcohol consumption until an arbitrary age. The French are famous for allowing their youngsters to consume diluted alcohols in the context of a family meal. Does that upbringing have an effect on alcoholism in the French compared to other nationalities?
Look, that's hard to say. I mean, there's a lot of individual difference. So I think that having a positive relationship with alcohol is generally good. So if you grow up in an environment where you consume some alcohol, but in a positive way, the people around you don't over-consume alcohol, and the relationship that you build with it changes. So then, when you do get older, you're less likely to abuse alcohol. But I don't know if there have been formal studies that have been done on that. But I do think that, I do know that the relationship with alcohol and the relationship that your family, the people closely surrounding you, that they have with alcohol impacts your relationship with alcohol downstream.
This lady says, CART also regulates food intake. Did you find any effects of the CART inhibition on food intake or body weight following the operant behavior testing?
So we didn't actually test food specifically, but we tested sugar, which has calories in it as well, just like food does. So we didn't see that it reduced their sucrose consumption, or actually we did test food consumption in their home cage. We didn't make them press a lever to get food. They could just consume food, and we just measured how much they ate. It didn't decrease that. It didn't decrease their body weight or anything else specifically. The only effects that we saw were really around that interaction between stress and alcohol. It didn't reduce motivation for alcohol without stress. It didn't reduce consumption of sugar when they were stressed. It had to be this combination between alcohol and stress before we really saw that it came into play. So really what we think is that, when the brain is changing after all these heavy drinking bouts, that this cart system might be adapting with the brain, specifically with that alcohol. And then what we're doing when we inhibit this cart chemical is that we're realigning it back to how it should be. But obviously very, preliminary studies, so we need to do further research.
Okay. One final question. Is there a link between Parkinson's disease and alcohol consumption?
I'm not sure exactly about Parkinson's disease, but yes, there is a link between cognitive decline. So alcohol-induced dementia is really a really interesting and important research line as well. There is projects in our lab with Professor Andrew Lawrence and Dr. Christina Perry, looking at the impacts of long-term alcohol consumption on memory and ability. So we know that that chronic alcohol consumption changes the brain and can impact your memory, and can increase your chances of developing these neurological disorders as well.
Thank you very much, Leigh, for a fantastic presentation. Thank you.